Hi everyone, this is the standardisation training on separate science biology paper 1 from the summer exams. In terms of what the session will look like, I will share a high tariff question with you. You will find that question in your pack and have a go at marking it. We will then discuss marking in your groups. I will then go through how many marks were awarded for that question last summer and what those marks were for. And we will then have another go at that same high tariff question to get some additional practice. If you do have any questions, my email address is at the bottom of that slide. Please do email me with any questions that you might have based on what we look at today. One thing to mention is that not everyone will agree all of the time. This is often the case in the summer when examiners are marking, but remember the purpose of standardisation is to ensure greater consistency in our marking and hopefully align to the marking criteria within our departments. In today's session then, we will review some student responses from last summer, which will be useful for us. If you are working in groups, you will be able to compare your marking with other people. If not, you can still compare your marking with the examiner from last summer and see how they compare. We will gain a deeper understanding of the mark scheme, especially with regards to some of the higher tariff questions. That should improve consistency within your departments in terms of your marking. And it should also allow for you to forecast and predict student grades a little bit better when we see what different levelled responses look like. Some general guidance then, first of all. Remember to read the examiner's reports and annotate your mark scheme with anything that you find that is useful. Those examiner reports have lots of insight into common mistakes and misconceptions, so identifying those and then thinking about them when you are marking will really help to support with that. Completing the training today ensures that we've had the opportunity to mark some of those high tariff questions before we get stuck into our mock marking. Anonymise your student scripts. It's easy for bias to creep in when you know the students whose work you are marking, so anonymise those scripts to try and alleviate that. Mark those high tariff questions first and mark them in blocks. High tariff questions are going to have the highest cognitive demand on you as a marker, so if we leave them until last, it can lead to mistakes. By marking them in blocks, you can comparatively mark your students' responses, which again is going to make your marking a little bit more consistent. Any content that is in brackets on the mark scheme is useful information for the student to have mentioned, but not essential for them to gain the mark. And finally, unless words are underlined, phonetic spelling is allowed, including the jumbling of letters. So for example, vacuolo is creditworthy, even though it doesn't sound like vacuole, it's clear what the student was trying to get at, so that would be creditworthy. The first of our high tariff questions then is this 6 marker on food testing. Using a red pen, underline aspects of the answer that you feel are creditworthy and decide how many marks in total you would have awarded. If you are in a team, discuss that as well, and remember to write the number of marks that you have awarded in the box at the bottom. Unpause the video when you are ready to move on. This response was awarded 3 marks in the summer. The student did correctly identify the iodine test for starch, as well as identifying the starting colour and the positive colour for that test. Remember that 3 points doesn't always equate to 3 marks. We need to be using those level descriptors. To help you with this, there is a little more guidance that we can use with regards to those descriptors. To get into level 1, a student must have some relevant content from any of those blocks on the mark scheme. To get into level 2, the student needs to correctly identify one of the reagents and the associated positive colour. Our student response here, we can see that that student has done that correctly for their starch test. But they also need something additional in their answer to gain level 2. This student, because they have given the starting colour of iodine solution, have managed to access that level 2 bracket. To get into level 3, the descriptor mentions that the student needs to be able to produce valid outcomes from their response. Quite a lot is needed to get into level 3. Both reagents and positive colours should have been identified. The student must refer to using heat for that Benedict's test. It is worth noting, heat is necessary. The student also could have referred to boiling the solution. But if they have mentioned a specific temperature, that temperature must be at least 60 degrees Celsius. So if a lower temperature than that is mentioned, the student would lose that part of their answer. Iodine must be implied to be a solution. So if the student hasn't explicitly stated that, that is okay, but we would expect to see something like use 3 drops of iodine or using a pipette add iodine to the sample. If it is not clear that iodine is in solution, again, that student cannot access level 3. The final thing that is needed to get a level 3 response is a risk and a linked control measure. Risks identified here have to be specific to the practical. So you can see there the 3 risks that are creditworthy by AQA. Generic risks such as tucking stools under, standing up behind chairs, those things would not be creditworthy in these responses. Remember, the student has to have a linked control measure, so these points underneath each risk are what we are looking for there. Students cannot mix and match the risks and control measures. They must come from the same area of that mark scheme. The examiner report kind of highlights some of these things. So as we can see, Benedict's needs to be heated, iodine needs to be in solution. And the main issue that you are likely to face is that students don't give a good enough risk assessment. So they often lack detail in the summer, and very few students correctly identified a hazard specific to these tests and didn't give those valid control measures in addition to that. Now that we have looked at one, we'll take a look at the next slide. Another student response to that same question then. Spend some time underlining in red aspects of that answer that you feel are worth a mark, and decide how many marks in total you would have awarded. Use that information from the previous slide to support with this, and unpause the video when you are ready to move on. This response then was deemed a good level 2 response, so it achieved 4 marks in the summer. In terms of where those marks came from, the student correctly identified their starch test along with the negative and positive colours. The student also mentioned the Benedict's test, that they would heat that to an appropriate temperature, and the starting colour and final colour for that test. However, what let this answer down was that final paragraph. There is no specific hazard or control measure that is mentioned in this answer, and unfortunately that limited this response. They did mention both reagents and those positive colours. They did mention using heat. They have made it clear that iodine is a solution. They've used the term drop. However, because of the lack of risk and control measure, this student has a good level 2 response but can't unfortunately access that level 3 bracket. Take any notes that you feel you still need, and then we will move on to the next question. Next high tariff question then is a 5 marker on starch testing in leaves that have been left in different conditions. Spend some time underlining in red parts of the answer that are creditworthy and decide how many marks you would have awarded. Unpause the video when you are ready to move on. This answer was awarded 2 marks in the summer by the examiner. The first marking point that was awarded was for specifying leaf 1, no chloroplast exposed to light, so no photosynthesis could occur. This is marking point 1 on the mark scheme. It is worth being aware of what is needed to get that mark. The student needs to specify the leaf, the factor that is missing, and the effect on photosynthesis. This student then does the same for leaf 2, correctly identifying the factor that was missing and the effect on photosynthesis, meaning that they also were awarded marking point 2. Marking point 3 is for referring to leaf 3, photosynthesis taking place, and the reason why. This student does not get this mark as they have not specified the factors that were present in order for photosynthesis to take place. It is worth noting though that if this student had have said the leaf has everything it needs for photosynthesis, they would have been awarded marking point 3 because previously in their answer they have specified the factors for marking points 1 and 2. Marking points 4 and 5 are then for referring to the production of glucose and the production of starch. This student does not mention glucose anywhere in their answer. They do make reference to starch by saying that all starch was used by leaf 1 to keep the plant alive. But because there is no reference to glucose, the student can't get marking point 5. Notice that there is a fallback mark, so if neither of those marking points have been awarded, starch being broken down can get 1 marking point. Because this is the fallback mark, it does need to be really explicit and clear what the student is referring to. The examiner in the summer didn't feel that that was the case, so the fallback mark wasn't awarded. A couple of additional things from the examiner report that are worth highlighting. Students must refer to photosynthesis in absolutes. So less photosynthesis or slower photosynthesis would not be creditworthy. And if a student refers to carbon dioxide in symbol form, it must be denoted correctly. You can see some examples there of incorrect symbols that would not be awarded the marking points. Take any notes that you need from this slide, and then we'll move on to look at a second example. Here is the second response then for this question. Underline in red things that are creditworthy and decide how many marks you would have awarded. Unpause the video when you are ready to move on. This response was awarded 2 marks in the summer by the examiner. The student correctly identifies that in leaf 1, photosynthesis wasn't able to occur as light was blocked. The student does not correctly refer to leaf 2 or 3 in terms of factors, but the final part of their answer does refer to glucose, so we need to take a look at marking points 4 and 5. The student says that for leaf 3, excess glucose not used in respiration is stored as starch. We can see that converse points are allowed here, so the student is on track to get marking point 5. They haven't used the word convert, but they have said stored as starch, and this is creditworthy. A good tip here is to look out for carbon dioxide. That is nowhere in this student's response, so immediately we know that this student cannot be awarded marking points 2 and 3. Take any notes that you feel you need, and then we'll move on to the next high tariff question. This question is a 6-mark magnification calculation. Underline in red parts of the student's answer that you feel are worth a mark, and decide how many marks you would have awarded. Unpause the video when you are ready to move on. This answer was awarded 3 marks in the summer by the AQA examiner. Marking point 1 was awarded. The student correctly used the information from the question and substituted the correct values into the equation that was given. The student also then obtained a correct answer from that calculation, which also gets the student the 2nd marking point. On the next line down in that student's working, they have correctly recalled the magnification equation, and this gets the student marking point 3. Whilst this was quite a straightforward answer, lots of the answers are a little bit more confusing than this one, so I'll just give a little more guidance in order to support you with the marking of that question. The first 2 marking points are for correctly substituting and then getting the correct answer from those values given in the question. If the student chose the wrong sum, for example forgetting to square 125, that would lose them both marking point 1 and 2. So there is no error carried forward for that marking point 2, which is really important to note. Marking point 3 is for recall of the equation. A triangle is not sufficient for this mark. The only exception to that is if the student has been awarded marking point 5 later on in their calculation. Marking point 5 is for correctly using their calculated values in the magnification equation. If they have done that, we know that the student does know that equation and how to use it. But if that mark is not there, a triangle on its own is not sufficient to get marking point 3. That is something I would stress in lessons when you are teaching that magnification topic. Marking point 4 is the conversion mark. This can be awarded anywhere in the student's answer, and there are 2 conversions that could have been completed. Most students will convert their millimetre reading into nanometres. However, the student could convert the nanometres into millimetres. This is their value from marking point 2. So it is fine if the student has converted that correctly from nanometres back into millimetres. Marking point 5 is for using their calculated values in the magnification equation. Notice that the student does need to use a correctly rounded value for this. If the student has incorrectly rounded their calculated value, that would mean that the student cannot access that marking point. The final marking point is for a correct answer. Notice on the additional guidance column, the student can give an answer that is consistent with either an incorrect rounding from the previous answer or from an incorrectly calculated value from previous in their answer. Because of these couple of error carried forward marks, it does mean that it's well worth having a calculator handy, as there are lots of possibilities that can still get some of that credit. A few key pieces of information from the examiner report as well. The first one being that any representation of pi, despite the question asking students to use 3.14, was creditworthy and were accepted. This again means that from the very off, really, there are multiple correct answers that students could be giving, and it's really important to have your calculators ready in order to check some of those. The other piece of information on the examiner report that I feel is just worth mentioning to you is that power of 10 errors, if there are no other mistakes in the answer, will usually mean that a student can get 5 marks. For example, if a student gives an answer of 801 or 8.01, they are likely to get 5 out of 6. The only mark that they have probably lost is marking point 4 because they have incorrectly converted somewhere in their answer. Still make sure that you are checking that answer, even if there is a power of 10 error on the final answer line, just to check that the student has obtained that from the correct place. If you need to take any notes from this slide, please feel free. It's also worth noting that there are some additional materials in the folders that have been shared with your curriculum leads, one of which is a document from AQA which shows lots of those possibilities depending on which representation of pi a student has used. So it's well worth taking a look and finding that sheet, as that can be really helpful when you are marking this question. We'll now look at another example to get a little bit more practice on this one. Take some time to underline this response, decide how many marks you would award, and unpause the video when you are ready to go through this one. This answer obtained 5 marks last summer. The first marking point that was awarded was for recalling the equation, so the student gets marking point 3 immediately. The student then substitutes their numbers from the question into that equation, and that therefore gets them marking point 1 as well. Marking point 2 is for having the correct answer to that equation. Notice that this answer is different to the one on the mark scheme, but that is because the student has used the pi button instead of 3.14 as instructed. Remember that this is allowed, so the student can get marking point 2 as well. The next line of the student's working shows a conversion. The student is converting their nanometers back into millimeters. However, they have done this incorrectly, as they haven't divided by 1,000 for a second time, and therefore we cannot award marking point 4. Marking point 5 is for correctly substituting their calculated values into the magnification equation. The student has done this correctly. Notice that they have rounded their answer from the previous step. They have correctly rounded that, so they are allowed marking point 5 because that error from earlier in their answer has been carried forward. The student has then worked out the answer to that calculation and has got 8.014. They have then rounded this to 8 and given their final answer as 8. The examiner in the summer deemed this appropriate in order to gain marking point 6, again from carrying that error forward. As we mentioned at the very beginning of the session, People will always see things slightly differently and might disagree with marking in places. This is one of those places for me. I probably wouldn't have awarded marking point 6, as the student's answer to their calculated equation is actually incorrect. If we do 4 divided by 0.499 on a calculator, we actually get 8.016. However, because it has been correctly rounded to 8, the examiner in the summer clearly thought that that was okay in order to get marking point 6. But again, it just shows how important it is to have your calculator with you so that you can be checking the steps, especially when there are so many possible combinations of how students could have arrived at an answer. Take any notes you feel you need for this one, and then we'll move on to our final question of today. This question was a 3-mark describe question on alveolar features. Underline the points that you feel are creditworthy, decide how many marks you would award, and unpause the video when you are ready. This answer was awarded 2 marks by the examiner last summer. Marking point 1 has been obtained from referring to the large surface area. It is worth noting that SA and SAV are common abbreviations and are therefore acceptable. Good blood supply, again, students can refer to there being a large capillary network, many capillaries, or surrounded by capillaries. Blood supply on its own is not enough, as all parts of the body have a blood supply. The final marking point is for referring to the thin walls of the alveoli. This student does not get this marking point as they have not referred to the walls directly. If the word wall is not in the student's response, they are not going to obtain marking point 3. Lots of students fell down on this in the summer exams. From the examiner reports, a couple of things to also mention. The command word was describe. Lots of explanations were given. Please ignore these. They are not necessary for the marks. Does not matter if they are correct or incorrect. We are ignoring those. The other point to mention is that we are reading this answer as continuous prose. So even though we have been given those numbered answer lines, read it as a whole piece of text. The student could get 2 of their marking points in one of their answer lines, for example. Take any notes you feel you need from this slide, and then we will look at our last example for this question. For the final time then, underline in red parts of the answer that you feel are creditworthy and decide how many marks you would award for this answer. Unpause the video when you are ready. This answer was awarded 2 marks as well. The student refers to a good blood supply, specifically from the capillaries, so they have definitely got marking point 2. The student also refers to a large surface area, which gets them marking point 1. The student cannot get marking point 3 because they have not referred to the walls of the alveoli. They have mentioned 1 cell thick, and they have got the correct explanation of a shorter diffusion pathway, but remember we are ignoring that because wall is not present. Marking point 3 cannot be awarded. One final thing to draw your attention to is the fact that moist is not creditworthy. It's still present in lots of student revision guides, student websites, so just make a real point when you are teaching about the lungs that moist walls and moist lining is not accepted by AQA and is very unlikely to be creditworthy. Take any notes you feel you need. That is our final question that we are looking at. Thank you for your time. I hope that you have found looking at some of those examples useful. Remember, if you do have any questions, email me. My email address is at the bottom of that slide there. There are some additional student responses on your printouts that can be used as additional practice if required. Again, feel free to email if you have any questions about those. Thank you.